So we were discuss actually discussing these transferases. So the subclass 1 denoting the 1 carbon transfer, 2, 3, 4, 5, 6, and then finally seventh subclass in this transferases is called phosphotransferases. So these are called kinases, for example. Similarly, each enzyme is given a name. So phosphotransferases are kinases. Similarly, the transferase is called aminotransferases. Sulfotransferase is called dehydro. Okay, so this is apiandrosterone sulfotransferases. Okay, so this is the one name, and second is choline sulfokinase. So kinase can also be given to sulfotransferases, but some specific enzymes only having this name. Okay. Similarly, for different transferases, for different subclasses, there is a specific enzyme: acetaldehyde transferase, aminoacyl transferase, permeryl transferase. Okay, so like this. Okay, fine. So let me move to Pathfinder. So this is given here also. So this particular number is for hexokinase, and these numbers are respectively showing the class, subclass, sub-subclass, and the fourth number is a serial number, that is 2 here, for example, indicating the order in which each enzyme is added to the list. So this particular number is showing, this is a serial number, so it is showing in which order the enzyme is added to this particular EC number. So this is a specific EC number serial. Okay, so in that particular category, the enzyme is added in that particular order. Okay, so this is given for one new enzyme is discovered, so one new number is given. So it will add, one will be added. Okay, so now okay, so one thing I wish to show you, for example, the first class that is called oxidoreductases is able to do some oxidation reduction reaction. So for example, there are two substrates, one is called A substrate, one is called B substrate. So this will be reduced. This is a reducing agent, this is the oxidizing agent. So finally this will be oxidized, this will be reduced like this. So one will be oxidized, one will be reduced. So these are called redox reactions. So these are involved in the oxidation reduction reactions. Type of enzyme which are involved in this category. So sometimes in the competitive examinations, you were provided with these enzyme names. For example, oxidase, peroxidase, oxygenase, dehydrogenase. So you are very familiar with dehydrogenases, but oxidases, peroxidases, and oxygenases, these are also involved in oxidation reduction reactions, and all these are examples of class 1 that are oxidoreductases actually. For example, oxidases are using oxygen as an electron acceptor. So what is the difference between oxidase and oxygenase? So if the enzyme is using the oxygen as electron acceptor, but do not incorporate the electron into the oxygen, okay, so this is called oxidase. But directly incorporates the oxygen into the substrate, then this is called oxygenase. Then there are some dehydrogenases, for example, alcohol dehydrogenase. Okay, so this is response lactate dehydrogenase. So these use molecules other than oxygen. So the substrate will be varying according to the name of that particular enzyme. For example, lactate dehydrogenase will use lactate instead of oxygen. Alcohol dehydrogenase will use the pyruvate or acetaldehyde to generate the alcohol. So, and also some cofactor, for example, NAD positive as an electron acceptor. Okay, so the NAD positive will accept the electron and it will be converted into NADH. So you have seen many cofactors. For example, NAD will be converted into NADH plus H positive, FAD will be converted into FADH2 in many dehydrogenase reactions. Also, peroxidase is using this H2O2 as an electron acceptor. So depending upon this electron acceptor, there are different categories of enzymes, and all are coming under the same class, that are oxidoreductase. Then I'm interested in transferases. So transferases are involving these kinases, transaminases because they are transferring the amino groups, transcarboxylases, they are transferring the carboxylate groups or CO2 group. Okay, so all these are coming under the. So there is a two substrate. One is so A is already linked with B, and there is other substrate that is C. So if this B is transferred to some other substrate C and A will be remaining, so this type of reactions are catalyzed by these transferases. They will transfer the groups to other substrates. For example, substrate C here. Okay, so next class is hydrolases. Hydrolases are a type of enzymes which will cleave the bond and hydrolase. And this hydrolysis reactions is catalyzed by adding the water molecule, because the water molecule or hydrolysis will generate as first an OH negative, and then the bond is finally broken down. So, for example, this is a substrate AB, and if the addition of water molecule to this particular substrate will lead to the generation of H positive and OH negative, and thus the two different products will be formed. So now the question is, which type of enzymes are coming under the hydrolase class? So there are phosphodiesterases. You know these phosphodiesterases, we have discussed them in cell signaling class. So these are responsible for breaking the cyclic nature of the secondary messenger molecules, for example, cyclic AMP and cyclic GMP, to convert them into non-cyclic forms. So cleavage the phosphodiester bond. Any phosphodiester bond they can cleave. Phosphatases, you know, actually phosphatases are opposite of kinases, so they remove the phosphate from the substrate by this hydrolysis reaction. There are peptidases also, lipases also, so lots of enzymes are coming under this class. So, okay, so if someone want to add more enzymes in this particular category, so you can note down some names. Actually, I have written from somewhere. So, okay, so I'm writing here. So, for example, alkaline phosphatase. Alkaline phosphatase is also a kind of hydrolase. Then there is, okay, so second is, for example, urease. So you should remember urease is was the first enzyme to be discovered and crystallized. Okay, so this urease is also a kind of hydrolase. Then there are peptidases. If someone asks you an indirect question, for example, trypsin. Okay, and pepsin, for example, pepsin, elastase. So all are the peptidases, carboxypeptidase. And you should remember all the peptidases which are able to degrade the proteins are coming under this particular class. Okay, so these are all peptidases which will cleave the protein. So they are also a lipase actually. So lipase is it can also come under the same category of this class. Okay. Then there are also, if you remember, lysozyme, okay, and some of the glycosidases are also coming under the same class, because they are also using the water, adding water to break the either glycosidic bond between the two sugars. Okay. So after this, I will move to other class that is called lyase. Lyase is a kind of enzyme that catalyzes the breaking of these bonds, but without hydrolysis. So it will not involve the water molecule, but instead of water or oxidation or hydrolysis will be not the case. So other than these two things, either hydrolysis or oxidation, so they can involve the elimination reaction. So it is written here very clearly. They have, okay, so they have given here already. So the elimination reaction and result in the formation of the double bond or a new ring. So, or conversely, they are adding group to the double bond. So it will be involving the double bond. Either they will break the double bond or they will create double bond or a ring system. So they're called lyase. For example, this is a double bond, and there is a other substrate that is having a functional group X. So this functional group will be added to this double bond, so that A will be forming a bond with the X, and the B will bond with other hydrogen. Or conversely, they can be added to either here A will form a bond with X, or B is having H, and this bond will be converted into a single bond. This is also possible. Okay. Now, what type of examples it have? So there are lots of enzymes in this class also. For example, aldolases. Okay, aldolases, synthases. So most of the synthases which do not require ATP or except a few, because some of the synthase which we are going to discuss in the last class also, but mostly the synthases are part of lyase. So you should remember this point. So aldolases, synthases, dehydratase, decarboxylase, all these enzymes are coming under this class. And I would like to add two more examples to this class so you can note them. So one is called DNA lyase. So this is not DNA lyase. And the second enzyme is ammonia lyase. So these lyases are also coming under this fourth class. Okay. Then we will move to next class, that is fifth class, also called isomerase. So isomerase are also important, and one important enzyme. Oh, sorry, I'm not able to scroll it. So isomerases are type of enzymes which will do some rearrangements in the same molecule. For example, some molecule like this, it will convert it in like this. So catalyze several type of intramolecular rearrangements. So if you remember this particular term, so nobody is going to question you again about isomerases. So they will do intramolecular. What is intra? Intra is the same molecule, the same molecule, intra. And yield the isomeric form. So what kind of enzymes are coming under the class? Mutases, cis trans isomers, isomerases, epimerases, racimases, and one more very important example is, okay, so writing here, fumarase. Oh, sorry, fumarase. So fumarase is enzyme which is coming under this isomerase class. So it will convert, if you do not remember the Krebs cycle, this fumarase is responsible for converting the one of the intermediates of a Krebs cycle into another. So generally, if you move through the Krebs cycle, you will find the fumarase. Okay, so specifically, so it is a kind of isomerase, and it will convert your fumarate into malate. Okay, so also you need to remember this from here. So it will convert fumarate into malate. Okay, so in the Krebs cycle, so this fumarase is also an example of isomerase class. Then there are last class is called ligases, which will add the two molecules by forming these bonds, but very important, they will need some of the energy molecule cofactors. So these energy molecules are either ATP, hydrolysis of ATP, or other common cofactor, for example, NAD positive, NADH, or NADPH, or like this, FADH2. So these include synthetases. So synthetases will require ATP, and they will make a bond between the two substrate A and B to make this product. Okay, and with the release of some ADP, low energy molecule. Examples are carboxylases. So they will use CO2 as a substrate. Pyruvate carboxylase, for example, so it will add the pyruvate or carboxyl CO2 directly to the pyruvate, and it will generate a 4-carbon compound, oxaloacetate or something like this. So also there are other enzymes. Okay, so one important thing actually I want to discuss here. So you need to note one name that is called tryptophan synthase. So actually, I also so tryptophan is an amino acid. So the synthesis reaction of tryptophan. So this is denoted by W. Okay. So tryptophan synthase is not synthetase. Actually, this is synthase, but it will be included under the ligase class because this will use some high energy cofactor, and Okay, so if you do not remember the amino acid biosynthesis pathway, so let me move to another PDF. Okay, so I hope this one. So this synthase that is called tryptophan biosynthesis pathway. Okay, so this is synthesis of tryptophan. Okay, so if you remember this pathway, so this phosphate is getting converted into anthranilate with the help of some enzyme, anthranilate synthase, which will convert glutamine into glutamate and transfer the amino group to this particular molecule phosphate to make anthranilate. Then this will be converted into N5 prime, some intermediate, that is an intermediate of tryptophan. Then finally this tryptophan synthase, which is an enzyme which is using some molecule called serine. So it will finally convert indole 3 glycerol phosphate directly into tryptophan with the release of this water molecule. So water is being released from this particular reaction. Also this PLP, that is pyridoxal phosphate, which is coming from vitamin B6, is required so that the serine molecule can be transferred over this particular uh, ring system. Okay. So this particular reaction, this tryptophan synthase, is using some serine residue to synthesize another amino acid that is called tryptophan, and water is generated in this particular reaction. Okay. Now we will move back to, sorry. Okay, so these are actually 6 classes, but recently one more class that is called 7th class is being added into this particular classification. So the 7th class is completely different from these enzymes, whatever we have discussed in this particular last table. Here in the 7th class, they have included some translocases. So you know translocases? These are the enzymes which are used to transfer. Okay, so there is a query also. Okay, so let me show you something. Okay, so actually this, you have noted this one actually. This is one, actually in your Pathfinder it is given 2.7.1.2 I think. Okay, it is so. Yeah. So actually this last number is a serial number. So this serial number is for the enzyme which is being discovered in the sequence in this particular class. Okay, class, subclass and subclass. If everything is matched, then this number is given for, okay, so hexokinase can be 2 types. Okay, so either glucokinase, for example, or journal hexokinase. So if it is a journal hexokinase, it is written like 2. Okay. And if it is a glucotransferase, means it is transferring the group to the glucose specifically. So if you remember from the Leninger, it is written here, the 4th number 1 is denoting, this 1 is for D-glucose as a phosphoryl group acceptor. So remember this thing. So firstly, this D-glucose was found to be acceptor, so this number was given. Then some scientists discovered that this hexokinase can be without specificity, without much specificity, can transfer this phosphate group to any hexose, either mannose or, okay, so either glucose or mannose. So actually that was given the second number. So this is not different. So the last number, if the last number is varying, so this is not a very much difference. But if you find something, so if you move in this direction, this class number matters a lot. If the number, this number changes, it means everything, every property of that particular enzyme will be changed. If this changes, this is a subclass of the same class. So it is also affecting lots of properties, because we know either one enzyme with this subclass can transfer the amino group, the second enzyme can transfer the phosphate or sulfur group. So this also matters a lot. But this is a subclass, and this last number matters very least. Okay, so if this is varying, it only means that the acceptor is different. Okay, so and it is discovered after that particular enzyme. So this 2.7.1.1 is discovered first. Then scientists discovered the 2.7.1.2. That is also a hexokinase. You can call it hexokinase, or you can call this particular enzyme as glucokinase, because this is transferring this. In this case, this number is denoting this D-glucose is the phosphoryl group acceptor, specifically the glucose. Fine. So who asked this query actually? Nikita, are you able to understand this? Okay, fine. So the last number, it is least significant actually. So it is only showing the order of discovery, but everything is decided by these three numbers. So now we will discuss some more examples, but let me quickly finish the this thing. So the seventh number or seventh class number is given to some specific enzymes which are called translocases. So if you know translocases, these are the enzymes which are required. So we know all the six classes, then how it has become apparent that none of these could describe the important group of enzymes that catalyze the movement of ions or molecules across the membranes and their separation within the membranes. So actually, scientists have classified different type of enzymes and almost each type of enzyme in these six classes, and then they feel like there is another category of enzymes, for example, which catalyze the movement of ions or molecules across the plasma membrane, and actually we cannot classify them in these six classes, so they make a new class with the international agreement where, so all the biochemists from around the world or globe or different molecular biologists have again made one more class. So these are called translocases. So the reactions catalyzed are designated as transfer from side 1 to side 2. And this side 1, okay, now there are two classes, sorry, these are two subclasses. Either the translocase, so this is a class, subclass can be based upon whether this particular enzyme is transferring the molecule or some ion from cytosol to extracellular phase, or from extracellular side into the cytosol of the cell. So based on this side 1 or side 2 transfers, so they can be classified further. Subclass in this EC7 designated type of ion molecules translocated. Also, the subclasses in this particular class are designated depending upon which type of ion or molecule is being translocated across the plasma membrane, whether a steroid molecule, whether a ion, or some other type of molecule. So you know aquaporins. So aquaporins are kind of translocases. So they will transfer the water molecules across the plasma membrane. So before starting the how enzyme operate finally, so let me quickly also tell you about the other subclasses which are given in this particular PDF. So today I will share this particular PDF with you all. Yesterday I did not share with you all because actually we did not discuss this in detail. So actually first table is showing about the type of reactions which we have already discussed from the Pathfinder, also from the Leninger. So, and these reactions are given here. So if you see these reactions, so you will able to understand what actually they are doing. So, okay. Then, so for example, we were discussing this lyases. The lyases are having some AXBY. So a double bond will be generated between A and X. And then finally, the Y group will be transferred from this to this one. This can also happen. So anything can happen, but you need to remember it will always involve either the formation of a double bond or the breakage of the double bond to convert it into a single bond with some elimination reactions. Okay. So after you are able to understand this specific table that is showing the classes, then let me show you the subclasses. So these are subclasses of these classes. These are the examples of some common enzymes, and these are the reaction catalyzed by these enzymes. So I will tell you which one are the most important among these enzymes. So first of all, so all the dehydrogenases are coming under this class that you know. Uh, oxygenases, peroxidases are also coming under the same class, peroxidase, oxygenase. Okay, so peroxidase, one example is catalase, and the kcat number is anything that is the maximum number of molecules an enzyme can convert into the product per second. Okay, so which can be converted into the product within the specific time. So catalase is having the maximum kcat number actually. So the catalytic power of peroxidase is found to be maximum. Then there are phenylalanine, hydroxylase, tryptophan, oxygenase, and all these enzymes which are having oxygenases, oxidases, and dehydrogenases. They are coming under the same class, oxidoreductase. And these are the subclasses. So if the 1.1 is given to some dehydrogenases, for example, lactate dehydrogenase, malate dehydrogenase, the second is given to some oxidase. So 1.2, 1.3, 1.4, 1.5, 1.6 oxidative deaminases, so which are used in amino acid oxidases, liberation of some ammonium ions, NS3. Then there is a second class that is transferases, which we have already discussed. So I'm skipping this and directly leading you to third class. Third class is hydrolase. So these are having esterases, peptidases. Okay, so peptidases we have discussed, pepsin, elastase, trypsin. All these are peptidases. Glycosidases, for example, lysozyme. These are very important actually. So these are coming under this particular class. So you need to remember lysozyme and trypsin are and acetylcholinesterase, for example, which is involved in the nerve transmission. So these are also kind of hydrolases which will use hydrolysis reaction to break down the specific molecules, like the hydrolysis of esters, peptide bond, glycosidic bonds, and so on. Then there are other enzymes also which are sorry. So some phosphatase, for example, ALP is an example. Okay, some deaminases which will remove the amine group, either glutaminase. So these are important actually. So you need to remember this also. So if you are having glutaminase, and then you can predict the second one that is called asparaginase also is also coming under the same class. So here you can write asparaginase. So asparaginase, glutaminase, all are coming under these deaminases. Then there is deamidases, which will, for example, nicotinamide deaminase, which will hydrolyze the amide bonds or CONH bonds. Okay, then there are lyase, which will lyse either C double bond C. So it is first subclass, second subclass is C double bond O lyse and C double bond N lyse. So they are having aldolases, DNA lyase, and ammonia lyase, which we have discussed in the table actually. So you need to remember from this particular thing actually. This aldolase is important. So you need to remember aldolase is a kind of lyase which will degrade the C double bond C. Then there are isomerases, epimerases, transracemase. This all we have discussed. So fumarase, if someone asks you, fumarase is a kind of cis-trans isomerase. So isomerases, and we know from the previous discussion, this fumarases need to convert into conversion of geometrical isomers. For example, fumarase and malate. So okay. Finally, there are some ligases which will ligate these bonds either subclass 1, 2, 3, 4. So they are tyrosine RNA ligase, acetate coenzyme, tryptophan synthase is also coming under this class. And there is pyruvate carboxylase, PC enzyme, which is also involved in the formation of this bond. Okay. So finally, we have finished this also. You need to remember few examples at least, okay, so that you are able to uh, answer if someone asks you a direct question. Okay, so epimerases, racemases, they are showing that they are isomerases, but here at least you should know this because the tryptophan synthase is very important because from the name it is actually synthase, but actually this is an example of ligase which is using some kind of cofactor and lead to the synthesis of water molecule. Acetate coenzyme ligase, tyrosine RNA ligase, pyruvate carboxylase also. So these are all ligases. So obviously they will use some of the cofactors, either ATP or some high energy molecule to catalyze these reactions to ligate two molecules. So this we are able to understand how these classes and subclasses are working. Then we are moving back to the. Okay, so we are moving to. So okay, so yesterday also I told you. So this is an enzyme or the catalyst, and enzyme is having a specific pocket where it is able to bind the substrate specifically. And this pocket or the substrate binding site is surrounded by some of the active site amino acid residues, which are part of this big enzyme and are involved in the catalysis of that particular reaction so that the substrate can be converted into the products. Okay. So I'm giving you a 2-minute break before starting this particular topic, how enzyme are able to catalyze the reactions. So if you have some query from the previous discussion of enzyme commission numbers, either classes, subclasses, or sub-subclasses, you can ask, okay? So you can ask your queries. So everything is fine. I hope there are no queries, but if you have any, you may ask. Because the cell cytoplasm is an aqueous environment and the cell is having pH around neutral, that is 7.2 exactly for cytosol. So if any reaction is uncatalyzed or the enzyme is not involved in catalyzing that particular reaction, so obviously it will happen, the reaction will proceed. If it is favorable, it will proceed without the enzyme also. Okay, but why we need enzymes? Enzymes are required to increase the rate of reaction so that in less time, more number of substrate can be converted into the products. And many enzymes are able to enhance the rate of reaction several fold. Actually, okay, so I will discuss some of the enzymes which are able to increase the rate of reaction or enhance the rate of reaction uh, unexpectedly. So there are few examples, but it is true for all the enzymes that they are able to increase the rate of catalysis so that these reactions can happen within no time. So enzymatic catalysis of reactions is essential to the living system. We know this. We need these enzymes so that we can catalyze some of the metabolic reactions so that we can get energy at least. And this is the second important thing for life to sustain life. after reproduction, actually, we need energy and we need to catalyze most of the biochemical reactions within our body or within our cells, so that we require enzymes. Okay, we need catalysts, biocatalysts. So these enzymes are also called biocatalysts because they are present in living systems. Under biologically relevant conditions, uncatalyzed reactions tend to be slow. Most biological molecules are quite stable in neutral pH, mild temperature, and aqueous environment inside the cell, because our cells are having this kind of atmosphere. There are aqueous environment, the neutral pH is neutral around 7.2, their temperature is also mild, so everything is fine. But without catalysis, these reactions will tend to slow. Now the question is, okay, so okay, so we are moving to the next topic. Okay, so here. So in cellular environments, actually many common chemical processes are unfavorable. So they cannot happen in aqueous environments of the cell, such as the transient formation of unstable charged intermediates or the collision between the two molecules in precise orientation required for the reaction. Because whenever, okay, so the enzyme will enhance the rate of reaction in two ways. What are these two ways? So first of all, they will hold the substrate molecules and will let them collide in a specific orientation, in a precise orientation, so that they can lead to the formation of products. Also, they will decrease the activation energy of the reaction, so that more and more number of substrate can be converted into the product. So this is a graph which is showing for the uncatalyzed reaction. So obviously this is a favorable reaction because products are having less free energy than substrate. But this process will, this chemical reaction will proceed through this transition state. So I will discuss what are the what is the basic definition of this transition state, what is the active site, what are the different intermediates. So we will discuss each term, no worries. So let me give you some basic idea how these enzymes are able to catalyze the reactions. This also has given in your Pathfinder. If you look into Pathfinders, they have given the two models. One is called lock and key model, one is called induced fit model. So we will discuss these also. But before starting this, so this is the lowering of activation energy. So with the help of enzyme, this transition state, okay, so this transition state will be having less energy, less free energy in comparison to uncatalyzed reaction. For example, this is the delta G or the free energy for uncatalyzed reaction. And this is the delta free energy for catalyzed reaction. So these enzyme will decrease the activation energy, so that so the activation energy or the EA is this particular energy which is required for the substrate to getting converted into the product. Okay. So, and how this is achieved? This is achieved by, because the enzyme is providing a local site for the substrate to bind. So if you remember this particular diagram, sorry, this, if you remember this particular diagram, so it is a large molecule, enzyme is a big molecule, but the some specific site or the active site can able to hold this particular substrate, so that it can separate the substrate from the cellular environment or the aqueous environment. So it will provide a local site or local environment where there is no influence of other proteins, no influence of other ions, no influence of other aqueous environment of the cell, so that substrate can be easily placed in an orientation, so that other substrate will bind to this particular substrate and will be converted into the product more efficiently. So that's how it is able to decrease the activation energy of the particular reaction. Now, So the two things will happen. Catalyst enhance the reaction rate by lowering this activation energy, this we know, okay, but you should also remember it will accelerate this interconversion, that is the substrate into the products, but there is no change in equilibrium point of the reaction. So the equilibrium of the reaction will remain fixed. Okay, so this question has been asked directly in the previous years of CSIR. So which one of the following will remain unchanged? So the equilibrium cannot be affected by an enzyme catalyzed reaction. So equilibrium will remain the same. Okay, now we will discuss everything, whatever is given in your Pathfinder also and also in your language. So the enzymes circumvent these problems. Okay, so by providing a specific environment in which the given reaction can occur more rapidly. It will bind the substrate at active site, and this will create a kind of environment where the substrate is free from all the influences of the cell cytosol. So two models has been proposed for this particular catalysis to happen actually. So first of all, okay, so okay, so an enzyme accelerate the rate of chemical reaction several times. This is very common. This we should know as compared to the uncatalyzed reaction in the water. So, okay, so let me tell you what are the different enzymes which can accelerate the reaction very. Okay, so there are different examples which are given in Manager also. So let me find 1 minute. So some rate enhancements which are a result of catalysis reactions are given in form of a table. Okay, I'm searching. It will also include your some of the common enzymes, some of the rare enzymes, and some of the very rare enzymes. So these are given in this table. This table is 6.5. That is some rate enhancements produced by an enzyme. So, for example, some cyclophilin can produce an enhancement of 10 to the power 5. So, it will increase the reaction rate by this fold. Carbonic anhydrase is also very efficient enzyme, and from the previous discussion, you should know this will use some metal cofactor, that is zinc ions, for the catalysis, and it will enhance the rate of reaction 10 to the power 7 fold, so that the carbon dioxide can be released out of the body. Then there is triose phosphate isomerase. It will also, this particular isomerase is actually used to convert this glycerol 3 phosphate in your glycolytic pathway. Okay. So if you remember the glycolysis pathway, so you will find one important enzyme, triose phosphate isomerase, which will be used to convert the fructose 1,6 bisphosphate into glycerol 3 phosphate and dihydroxyacetone phosphate. Okay, so this triose phosphate isomerase is converting these three carbon forms into each other, and it will increase the rate of reaction 10 to the power 9-fold. Carboxypeptidase A, 10 to the power 11, phosphoglucomutase, 10 to the power 12, and the most efficient enzyme are urease, and the better than this is orthidine monophosphate decarboxylase. This you have studied in nucleotide biosynthesis actually. So this was also discussed that if this particular enzyme is not present within the cell, it will take some million years to synthesize the DNA. Okay, so only with the help of this particular enzyme, orthidine monophosphate decarboxylase, we are able to create, we are able to produce de novo nucleotides at a very fast rate. So if someone asks you about which is the best enzyme to produce some of the rate enhancement, so it is always orotidine monophosphate decarboxylase. But if the question is something different, for example, about kcat value, so the kcat value is always the catalase is the answer. Okay, so there are two important questions we are discussing today. One is orotidine monophosphate decarboxylase, which will produce some rate enhancement, means it will increase the rate of reaction that much fold. Both are different things. And one more example is given, So it is triose phosphate isomerase. Actually, at the last page, I was discussing. So if you remember, triose phosphate isomerase is to convert glycerol 3 phosphate in dihydroxyacetone phosphate and vice versa. Okay, so this is a both way reaction which can proceed in either direction based upon the concentration of that particular substrate. So actually, I'm searching for the second table, which is showing some kcat values. Oh, sorry. So we will discuss all these metal catalyzed reactions, acid catalyzed reactions, and so on. I don't know what was that. We should have at least actually. Okay, so here it is given actually. This is turnover number or the kcat value. So turnover number is something. So the enzyme is able to catalyze the reaction and able to convert how many substrate into the product and some specific time. For example, it will be written somewhere. Okay, so the kcat number or the turnover number, if the question is like this, which enzyme is having the maximum turnover number? So the catalase actually, so catalase is catalyzing a reaction. It will convert H2O2 within cell because H2O2 is harmful to cell because it is an oxidizing agent. So it should be converted into some neutral form, that is water and oxygen, with the help of some peroxidase that is called catalase. So this catalase is also needing some metal ions, for example, iron Fe2 positive to catalyze this particular reaction. And C, this is per second. So this catalase will able to convert so many number of H2O2 into water and oxygen. So this is 4 into 10 to power 7. Okay, so also carbonic anhydrase is having a catalytic or the turnover number in the range of 4 into 10 to the power 5, and acetylcholinesterase 14,000, and so on. It will decrease actually. So the maximum is for catalase. So you should remember this particular enzyme because it is having the maximum kcat value or the turnover number per second. It will able to convert so many molecules of substrate into the product. And the second question is from the orotidine monophosphate decarboxylase, which is having the rate enhancement effect over the reaction is very, very high. Oh, I don't know. I have to move to some other chapter. Oh no. So fine. So actually, we were discussing. So two type of models have been proposed, and so also you should know these definitions. So the enzyme catalyzed reactions taking place within the confines of a pocket on the enzyme that is called active site. So active site, some specific site over the enzyme where it is able to bind specific substrate molecules. Then there is a molecule that is bound in the active site and acted upon by the enzyme. This is the definition of the substrate. So the molecule which is bound to the active site of an enzyme is called the substrate for that particular enzyme. Okay, and then this catalyzes the chemical transformation of this particular substrate, which is bound to the active site. Often the active site encloses the substrate, sequestering it completely from the solution. So this is also very important. So with this is not only able to bind it, it is sequestering, it is removing the substrate from the aqueous environment. And because we know in the cell there is different type of proteins, there are lots of protein within the cell cytoplasm. So they can interfere with the catalysis. So therefore, it will sequester the substrate from the solution. The enzyme substrate complex is an entity first proposed by, so this term enzyme substrate binding or the enzyme substrate complex is this particular term was given by the Woods in 1880s is central to the action of enzymes, and we know this type of intermediates are formed in each enzyme catalyzed reactions. So here, an enzyme molecule when binding with the substrate, it will remove the substrate from the solution. So, and product will be formed that is called enzyme substrate complex. This term was given by Woods in 1880s actually. So this is an intermediate. So this intermediate is first converted into another intermediate, and you should remember at any step this reaction can be reverted back. So this enzyme substrate complex will first be converted into enzyme product complex. Finally, these enzyme products are product is released. So you should remember from this particular discussion. So the enzyme is increasing the rate of reaction only. It is enhancing the reaction rate, but there is no change in the equilibrium. Equilibrium will not be changed. So the concentration of substrate and concentration of product will be not affected by the enzyme overall. And the second most important thing is, you see the enzyme is bonding with substrate and it is catalyzing this reaction by forming these intermediates. Finally, you will see the enzyme will not be converted. So एंजाइम के ऊपर कोई फर्क नहीं पड़ता है जब एक एंजाइम कैटलाइज्ड रिएक्शन होती है. So enzyme will not be modified in this particular reaction. This is very important point to note. So the enzyme will remain unchanged. Also the equilibrium of this particular reaction will remain unchanged. So now we are coming to two models actually, and before that actually, so enzymes are used in the process, but enzymes are not affected. Also, the equilibrium point is not affected. Now the question is how this is happening, how the proper orientation is achieved. So this actually given in your pathfinder very clearly. They have defining this. So formation of an enzyme substrate complex is the first step in any enzymatic catalysis. So the enzyme substrate complex should be formed. Without this, the catalysis cannot happen actually. Also, there are two queries. I do not know what you are. Is okay. So actually, I told you kcat is the turnover number. How to find kcat? Okay, we will come to that particular topic. Actually, this is the enzyme kinetics part. So because we are covering the enzyme introduction and basics first, and then we will start enzyme kinetics. And I also want to tell you that enzyme kinetics is a kind of approach to find the mechanism of enzyme action, or because we know some of the inhibitors types, competitive, uncompetitive, non-competitive, or mixed types. Based on these kinetics models, actually, we are able to define what kind of inhibition, enzyme inhibition it is. So that's why we will cover these particular points. So how to calculate the kcat value, what is the V maximum, what is Km value. So let me tell you Km, Vmax, what is this mm equation, what is mm plot, and then I will tell you for sure what is kcat value and how you will find this kcat value. Okay, so if you move to page number, for now, if you move to page number some 115 in your 7th edition Pathfinder 1. So the catalytic constant kcat value can be represented by K2. Okay, so I will tell you what is K2. And it is the V maximum divided by the total enzyme concentration. So if you want to calculate kcat value, so kcat, first of all, you should know this kcat is something is equal to K2. And I will tell you what is K2. And it is equal to V maximum. You know V maximum? V maximum is the maximal velocity can be achieved by this particular enzyme. And if it is divided by total enzyme concentration, so it is total enzyme concentration is like this. So this is total enzyme concentration is E, and this concentration is T. So this is K cat value actually. Now what is K2? In some cases actually you will find that it is simply equal to K2. So I will tell you about K2 also. So we had discussed one equation. Sorry, actually I need to remove this, otherwise I cannot move to the other area. So if you remember this particular equation, sorry it is. Okay, so this equation is showing, so this is K1 when this enzyme substrate complex is getting converted into ES, and this is K minus 1 when it is getting converted back. Also more clearly you can find from the Pathfinder also, it is given somewhere. So we will discuss all these things. What is the rate limiting step? What are the rate constants and all these things? No worries. Sorry. I need to cover so many things. Okay, this is actually kinetics is far away, so you need not worry actually right now. So suppose this is an equation. So these two scientists, one is Michaelis, one is Manton. So they devised this particular equation, and based on these equations, they are able to define these K1, K2. So if simply this enzyme substrate substrate converted into enzyme substrate complex. This is K1. If it is going back, this is K minus 1. It's similarly this ES is getting converted into enzyme and product. So this enzyme substrate complex is when converted into enzyme and product, it is releasing this product without changing in enzyme. So this is called K2. And mostly this will be unidirectional. So only in few cases this is found that this can revert back, but once this product is released, so this reaction is over. Okay, so we will discuss that point also. So this is pre-steady state actually, in which we are, and this is the equation where this V naught is equal to maximum velocity multiplied by substrate concentration Km plus substrate concentration. So you need to remember this also. Okay, for simplicity, you can find this particular equation. So based upon this equation, actually, most of the enzyme kinetics calculations are done. So one enzyme and substrate will be converted into enzyme substrate complex. So this intermediate form will be formed. And there is a very little gap between enzyme substrate, and you even do not know when this, within nanoseconds, actually, this enzyme substrate will be getting converted into enzyme product. This enzyme product complex will be degraded into enzyme, and this product will be released out of enzyme. So enzyme will remain unchanged. And generally this step after the enzyme substrate complex is getting converted into enzyme product after release of product, this reaction cannot be reverted back. So therefore this is called K2. So in some cases this K2 is defining the K cat value when this enzyme finally convert the substrate into the product. And how many substrate molecules can be converted into the product is defined as turnover number or K-cat value. Okay. So hope this is clear. Whoever has asked this part of the question. Ankita, are you able to got the point? Fine. So there is no worries. Also, if you are able to not get the point, we will discuss all these equations and the derivations in more detail in our upcoming classes, because this Enzymo or the advanced biocatalysis is a complex topic. It will not only involve the enzyme introduction, it will involve everything: enzyme classification, enzyme kinetics, inhibitions, and what type of reactions actually. So we are going to discuss these also. So general acid base catalysis, the second is metal ion catalysis, And all these things are also given in your Pathfinder also. So, but before switching to that particular page, I would like to tell you about these two basic models. One is lock model, lock and key model. One is induced fit model. You also have studied a lot in your previous classes about these two models. So I can skip, but I will not. Covalent catalysis, acid-base catalysis, and third one is metal ion catalysis. So these three basic type of catalysis mechanisms are shown by different classes of enzymes. So every enzyme is following one trend. Either it is a metal catalysis or some covalent catalysis or some acid-base catalysis reaction. So, okay, so let me quickly finish this before closing today's class. Okay, so the formation of enzyme substrate complex is requiring this active site. This active site is able to bind some specific substrate molecules. Once the specific substrate molecule is bound, the substrate bind to the active site of an enzyme by multiple weak non-covalent interactions. So someone can ask you this particular question. This is a basic question. So the type of interactions which are formed between the enzyme active site and the substrate are multiple bonds, and these are non-covalent interactions. For example, many kinds: hydrophobic interactions, hydrogen bonding, ionic interactions, or even in some cases, these reversible covalent bonds can also form between the enzyme and the substrate. The free energy released in the formation of a large number of weak interactions between the enzyme and the substrate is termed as binding energy. So, okay, so you got an term binding energy. So, what is binding energy? Whenever there is a free energy which is released in the formation of large number of weak interactions. So, these all weak interactions are formed. So, these weak interactions are formed as a result of some energy will be released between the enzyme and substrate, and this is called binding energy. The binding between the enzyme and the substrate is highly specific because they are specific kind of bonds formed between these two molecules. A given enzyme usually bind to only one kind of substrate, so they are very specific. Specificity of binding depend upon the precisely defining the arrangement of atoms within the active site. So one thing you should know, what type of amino acids or the atoms are present, so they will define the overall specificity of the enzyme. Some enzymes are very specific, some enzymes are not so much specific. For example, we have discussed hexokinases. Either it is glucokinase or hexokinase. Glucokinase is very specific for glucose, whereas hexokinase can bind the glucose or it can bind the mannose also. Whatever the case, but there is not much specific. Okay, so these interactions, these small interactions, these non-covalent interactions are defining these specificity of their particular enzyme. Binding energy is very important. Okay, then there is two important models. One is called lock and key model. Second is called induced fit model. Lock and key model do not assume anything. Everything is fixed. If the enzyme is having shape like this, the substrate will have shape something like this. If enzyme is having shape like this, the active site is like this, the substrate will be having shape like this so that it can bind with this more efficiently and specifically. Okay. Whereas induced fit is telling that so finally, it will bind, the enzyme substrate complex will be formed, and the enzyme will not change in this particular reaction, whereas this will be joined to make a product. So product will be released and the enzyme will remain as such. So this is lock and key model, which was proposed by Emil Fischer in 1894. Okay, so Fischer gave this lock and key model. So only you need to remember this. Fischer has provided this model. Then there is another model that is called induced fit model, which assume that the protein is having some conformational flexibility and they can change the shape of the active site when there is after binding of the substrate itself. So once the substrate bound loosely, then there is some conformational changes in the protein so that the substrate can interact more efficiently with that particular enzyme of the protein. Because most enzymes are protein only. The binding of the substrate induces a conformational change. Only after binding of the substrate, this conformational change is induced within this protein or enzyme so that it can bind the complementary fit once the substrate is already bound. So binding site has a different three-dimensional shape before the substrate is outlined. So see here, this is given a very clear pictorial explanation. So here, this picture showing this is having some shape like this, which is not very complementary to the substrate. So loosely bound to the substrate to make an enzyme substrate complex. But after this complex is being formed, so these shapes are more defined so that they can interact more efficiently with the substrate, so that the product can be released out of the enzyme and the enzyme will be restored back to the original conformation. So okay, so it will remain unchanged also in this particular case. Induced fit also do not say that enzyme will change. It will remain as such. After the reaction, you can find that enzyme. There is not a significant change in the enzyme. Only there is a conformational change during the reaction only. So enzyme lower the reaction activation energy, and this we have already discussed, so that this transition state can be achieved more efficiently. So the maximal binding energy is released when the enzyme facilitated the formation of transition state. So if this is a transition state, so this binding energy, maximal is binding energy is released because this enzyme is able to hold the substrate in specific orientation so that they can collide more efficiently. So okay, so enzyme binding energy released due to the interactions, these collisions between the substrate, interactions between the enzyme and the substrate, okay, lower the activation energy. Only the correct substrate can form the maximum interactions with the enzyme. So it is always like this, the who is going to make the maximum interactions. So, and substrate which is able to make maximum interactions, maximum non-covalent bond with the enzyme, that will be the specific one only. Okay, so that will be that particular substrate which is acted upon by the enzyme. एंजाइम उसी के ऊपर वर्क करेगा जो एंजाइम के साथ सबसे ज्यादा बॉन्डिंग नॉन कोवलेंट इंटरेक्शंस दो, बट एटलीस्ट some non-covalent interactions show करेगा. Thus the maximal binding energy is released because there are lots of interactions, so binding energy will be high. So when the enzyme facilitated the formation of transition state, so that this transition state can be overcome, so that the substrate can be converted into the products. So the transition state is the point of highest free energy. So if someone asks you what is the transition state is some point where there is a maximal free energy of the enzyme substrate complex is being formed. So that in which the reactants are partially converted into the product. Okay. So actually we are running out of time again, and we will discuss more about this in our next class. Okay, so for now I'm closing it here. So if you're having some queries, I'm unmuting you so that you can ask your queries at least. Okay.